Okay, so we've talked about monohybrid crosses and dihybrid crosses, and we know the patterns of inheritance that we expect to see with both of those. Now let's think about this with a little bit more relevance, right? Taking into account a little bit more of the complexity of the genome. So, for example, we know in humans we've got, not one or two genes, we've got 20,000 different genes in the genome. And if we're trying to think about how inheritance might happen for more than two genes, we can immediately see that it becomes immensely complicated. I mean, even if we took a very limited scenario where we're looking at, let's say, four genes A, B, C, and D in the genome, and I tell you that you're starting with two parents that are heterozygous at each of those genes, and they have an offspring, and I ask you, what are the offspring types that they could have? What's the likelihood that they would have any given offspring type over another? Well, as we talked about for our monohybrid and dihybrid crosses, you might think, well, step one, I need to figure out what types of gametes each of these parents could produce. And so if I do that in some kind of systematic way, I could say, all right, for this parent, I know either this dominant allele or recessive allele for A is going to go into each gamete, and combined with that, I'm going to have either the dominant allele or recessive allele for B, and then combined with that, either the dominant or recessive for C, and so on. And so we could actually do a little branch diagram to try and figure out all the possible combinations. So we've either got dominant A or recessive A going with either dominant B or recessive B, and so we'd have two branches for each of those, and then each of those possibilities would be combined with two branches, and we can immediately see the problem with this approach, right? Probably don't even need to go on and draw this out completely, but I will. What is the problem with this approach? Well, too many gamete types, right? It gets insane. If we've got two possible alleles at four different genes, we are going to end up with a 16 by 16 Punnett square. This is ridiculous. We can't do that. That would be way too many offspring to try and analyze in that fashion. So we are not going to take that approach of figuring out what the gamete types are and making a Punnett square. Instead, we're going to think about each of these genes, gene A, gene B, gene C, gene D, individually, and use probability to predict what types of offspring are possible with these two parents, right? And to figure out probabilities or likelihoods of any given offspring type. So let's talk a bit about probability. Um, a lot of students are intimidated by the idea of calculating probabilities, but it's really very approachable. You don't need to be intimidated at all. In terms of what a probability is describing, it's just describing the likelihood that an event is going to happen, right? That's all. The probability of something that's impossible is going to be 0, and the probability of something that is certain to happen is 1. So all your probabilities are going to lie between 0 and 1 and be some fraction thereof. So we're going to go through first a couple of examples that are going to simplify this a bit and then apply it to genetics, but let's lay down some ground rules here. So if we're thinking about probabilities of independent events happening together. So let's say I want event number 1 outcome and event number 2 outcome to happen at the same time or in the same individual or whatever. Then the way to calculate that overall probability is to take the individual probabilities of each event and multiply them with each other, right? Okay, so that probably doesn't make a whole lot of sense, but let's use an example of a coin flip. So if I flip a coin twice, what's the probability that I'm going to come up with a heads both times? So we know that for any given coin flip, you're either going to get a heads or a tails, right? So the probability on any coin flip of ending up with a heads is going to be 1 out of 2. You got two possibilities, heads or tails. Heads is one possibility out of those two, so 1/2 of coming up with a heads. But I'm saying I'm going to flip a coin twice. What's the probability that both times I come up with a heads? So heads and then another heads. We know that that second heads probability is also going to be a half. Since I'm saying I want this event to occur along with this event, what I'm going to do is multiply those individual probabilities against one another, and our overall probability of 2 coin flips giving me heads both times is 1/4. Okay, so that's what we would do if we were dealing with the probability of 2 independent events. This coin flip does not influence this coin flip. They're independent of one another. What's the overall probability of those 2 outcomes occurring together? It's the individual probabilities multiplied by one another. All right. Now, on the other hand, if we're trying to determine the probability of an event that can occur in 2 or more different ways, then we would need to add the individual probabilities. All right? So, for example, if I flip a coin twice, what's the probability of coming up with exactly 1 tails? So, exactly 1 tails out of 2 coin flips. Well, we'd have to recognize that I could either get that tails outcome on the first coin flip and then a heads. That would satisfy what the question is asking for. Or I could flip and get a heads on the first try and then a tails on the second try. Either one of these outcomes would give me what the question specifies. I flipped a coin twice and I ended up with exactly 1 tails. So to figure out the overall probability of that outcome, I would need to figure out the probability for this outcome and then add them together. And so just as we saw in the previous example, probability of a tails on any coin flip is going to be 1/2. Probability of a heads is going to be 1/2. So the probability of flipping a tails and then a heads is going to be 1/4. The probability of flipping a heads and then a tails, likewise, is going to be 1/4. And so because either this or this could happen and satisfy what the question is asking for, I'm going to add those up. And so 2 out of 4, or 1/2 of the time, I would satisfy that outcome. Right? Flipping a coin twice and coming up with exactly one tails. Now, what about this scenario? This is a bit more complicated. If I flip a coin 3 times, what's the probability of coming up with at least 1 tails? If I flip a coin 3 times, what's the probability of coming up with at least 1 tails? So, thing to recognize here is that when we see at least, that could mean coming up with 1 tails. It could mean coming up with 2 tails. It could mean coming up with 3 tails. All of those would satisfy at least 1, right? And so we could do this the long way. We could say, all right, I get a tails, and then I get a heads, and then I get a heads. I get a heads, and then I get a tails, and then I get a heads. I get a heads, and a tails. Or, look at all the ways that we could have 2 tails, which would also satisfy this, right? We could have tails, tails, heads, heads, tails, tails, or tails, heads, tails. That would also satisfy having at least 1 tails. And then finally, if we had tails, 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 that would also satisfy the outcome that was desired of at least 1 tails. So we could do it this way and figure out the individual probabilities for each of these outcomes. So the likelihood of having a tails and a heads is going to be 1/2 times 1/2 times 1/2, which is 1/8, right? All of these are going to be 1/8 all the way down. Same probability. And then we would do what? We would add them all up, because we're saying the first scenario or the second scenario or the second scenario or the first scenario or the second scenario or the third or the fourth or the fifth or the sixth or the seventh, all of these would satisfy the outcome of at least 1 tails. We would add those all up and we would see that 7 out of 8 times we would get that scenario, at least 1 tails on 3 coin flips. But the easier way to solve a problem that says at least 1 is to recognize that the only way that you wouldn't get at least 1 is if you got none. Right? If I flipped a coin 3 times and I got 3 heads, that's the only way that I wouldn't get at least 1 tails. This is a much easier problem to solve because I know that all of my events have to add up to 1, right? All of the possible outcomes for any possible event have to add up to 1. And so I can just figure out what the likelihood of getting 3 heads is. And we know that's 1/2 on each coin flip. And so overall, that's 1 out of 8. And then I know that all of my outcomes have to add up to 1, so I can just subtract that 1 out of 8 from 1, which leaves me 7/8. So 1 time out of 8, I would predict I'd get a heads and a heads and a heads. 7 times out of 8, I'd get something else, right? And the something else would have to satisfy my at least 1 tails. So this is definitely the quicker way to solve that kind of a probability problem, where you're asked for at least 1. But the key thing to note here is that if we're looking at the probability of an event or outcome that can occur multiple ways, we figure out the probability for each way and then add them up. Okay? All right, so how do we apply this then to genetics, and how can we use this to solve that kind of crazy problem that I outlined first? Well, if we're thinking about a diploid organism that has 2 possible allele types at a given gene, and let's say they are heterozygous, right? Let's say this is the S gene, and they are diploid, they're heterozygous, they have 2 alleles that are different. Whether they put a dominant allele or a recessive allele in any given gamete is the same as a coin flip, right? You have a 1/2 probability of inheriting the dominant allele, 1/2 probability of inheriting the recessive allele. And so we can use the same rules of probability to figure out potential gamete types if we are looking at genetic crosses, where we have different alleles segregating in the offspring. So let's say this is our smooth versus wrinkled offspring from our pea plant example we saw in the previous video, and we're crossing 2 heterozygous plants with one another. We know that those 2 plants are going to be producing gametes of the same type. Half of them are going to have the dominant allele, half of them are going to have the recessive allele. This parent will be producing the same gamete types because it has the same genotype as the other. And so if I want to know what is the probability of ending up in their offspring with this genotype, I just need to think about what's the probability of this parent passing on a dominant allele, what's the probability of this parent passing on a dominant allele. And I see that in either case it's 1/2, and so I would expect the probability of ending up with a homozygous dominant individual in that next generation as 1 out of 4. And we see that's exactly what we get, right? What's the probability of ending up with a homozygous recessive? Well, it's the same, right? We take the probability of ending up with a recessive allele coming from both parents. That's going to be 1/2 times 1/2, also 1/4. Now, what is the probability of ending up with a heterozygous individual? Well, here it gets a little bit more complicated because we have to recognize that what we're dealing with is 2 possible ways that that could happen, right? We have way number 1, in which this parent contributes a dominant allele and this parent contributes the recessive allele, and we know that that is going to be this sort of outcome, right? So parent 1 contributes the dominant allele, parent 2 contributes the recessive allele. But there's another way that we can get that, and that's if parent 1 contributes the recessive allele and parent 2 contributes the dominant allele. Now, in either case, the likelihood of inheriting a dominant allele from parent 1 is 1/2. The likelihood of inheriting a recessive allele from parent 2 is 1/2. So this outcome will be 1 out of 4. Same for this. 1 out of 4. And because there are 2 possible ways that could happen, we know we add those together and we get 2 out of 4, or 1/2 of our offspring that we expect to be heterozygous, and here they are, right? 2 different ways for that outcome to happen. So when we're thinking about more complex genetic problems, where we're combining multiple alleles from multiple genes, if we can visualize this Punnett square outcome of 1 out of 4 being homozygous dominant, 1 out of 4 being homozygous recessive, and 2 out of 4 being heterozygous, that provides a nice, easy visual snapshot that will help us analyze more complex problems like the one that I outlined previously, right? If we have this complex scenario where we've got all these different genes, 4 different genes, each of them having 2 different alleles, we can more easily figure out the types of offspring that we could get. So for example, if I asked you, what is the probability, P, of ending up with an offspring from these 2 parents that has this particular genotype? Homozygous recessive at the A gene, heterozygous at the B, homozygous dominant at the C gene, heterozygous at the D gene. What's the probability of ending up with that? All I would need to do is take each of these genotypes individually, figure out the probability of ending up with each individually, and then multiply them together, because I'm saying that they're happening in the same individual. So with these 2 parents, what's the likelihood of ending up with a homozygous recessive offspring? Well, we can model that using our Punnett square. Each parent is going to have a half probability of passing on the dominant or recessive allele, and so we put those gamete types on the outside of our Punnett square, and we draw our offspring types that we expect, just as we saw in our previous example. 1 out of 4 are going to be homozygous dominant, 1 out of 4 are going to be homozygous recessive, 2 out of 4 are going to be heterozygous. So what's the likelihood of ending up with homozygous recessive from these 2 parents? It's 1 out of 4. Likewise, what's the likelihood of ending up with a heterozygous offspring coming from these 2 parents? Well, again, we could visualize a Punnett square showing those 2 parents and the gamete types that they would produce, and it would be exactly the same as the previous example. A 1 to 2 to 2 to 1 ratio, so likelihood of a heterozygote coming from those 2 parents is 1/2. I'm not going to draw out the Punnett squares for C and D. You guys can do that on your own, but you would see that homozygous dominant, again, it's going to be 1 out of 4. Heterozygous, again, it's going to be 1 out of 2. And because we're saying we want this genotype in the same individual as this, as this, as this, right? So all of these happening together, we're going to multiply these individual probabilities, and what would we end up with? 1/4 times 1/2 is 1/8, times 1/4 is 1/32, times 1/2 is 1 out of 64. So if we had gametes randomly produced from these 2 parents, in every 1 out of 64 gametes we would expect to see, or 1 out of 64 fertilizations, sorry, we would expect to see an individual formed with this specific genotype. Right? So we can use probabilities to figure out more complex, higher order genetic crosses beyond a dihybrid cross without drawing out a crazy, gnarly 16 by 16 or even larger Punnett square. Okay. So you can expect to see those kinds of problems using probability to figure out offspring types. But let's move on and let's think about other types of inheritance patterns beyond what Mendel described. So these might be referred to as, beyond Mendelian genetics, stuff that he did not report on, but which we since have figured out definitely happen out there in nature. The first pattern that we're going to look at is incomplete dominance. So whereas Mendel described a pattern of complete dominance with all of the traits that he was looking at in pea plants, so purple completely dominant over white, white receding into the background completely in the hybrid, smooth completely dominant over wrinkled. There are many examples out there in nature of an incomplete form of dominance where one allele does not completely hide or mask the other when present in the heterozygous state. So for example, there's snapdragons that have true breeding strains in which the plants always produce red colored flowers and white colored flowers. And if the inheritance pattern observed here was the same as what Mendel described, if we cross these 2 true breeding strains, then what we would expect in the next generation is 100% of those F1 offspring to resemble the parent that has the dominant flower color. So for example, red. But if you cross red and white snapdragons that are true breeding for this flower color trait, what you see is not red offspring in the F1 generation. Instead, you see a pink flower color, which is intermediate between the two parent strains that we started with. So we have an intermediate phenotype for this F1 generation. Now we know what the genotype of this F1 generation has to be, right? Because we know the genotypes of these parental generations. If this is true breeding, we know it has to be homozygous. Let's just represent the red color allele with a capital R. These are likewise true breeding, but they're white. Let's represent that allele of this flower color gene with a lowercase r. 100% of these offspring from these two parents are going to have to be heterozygous, right? 100% heterozygous offspring. So in the hybrid, we don't see that this R allele, this red color allele coming from this red flowered plant, as completely masking the white phenotype. Instead, what we see is sort of a blending between the two, an intermediate phenotype where one allele of this gene is not quite enough to get us all the way to red flower color, but maybe it gets us halfway, so a faint red or pink flower color. So this is an example of incomplete dominance. Now, this sort of resembles what we might expect to see if inheritance was characterized by that blending pattern which Mendel disproved, right? So how do we know that this is not a blending pattern of inheritance? Well, let's think about what would we get in our next generation if we allowed individuals in this F1 generation to interbreed with one another. So we crossed this F1 generation. What would we get in the F2? So pause the video and make your predictions for what we would see in that next generation, and then I will draw it out. But it's worthwhile to go through the exercise of doing it yourself. All right, so what would we expect to see? Well, again, we're going to draw out the gamete types that each parent would produce here, and we know that they would be either the red color allele or white color allele coming from each parent, 50-50, right? In each case, we could represent those on the sides of a Punnett square, combine those in all possible combinations, and what we would expect to see is what we've seen before, a 1:2:1 genotype ratio. But what phenotypes would be associated with these genotypes? This individual that has two red alleles would be red. These two individuals that have hybrid genotype or heterozygous would both be pink, as we saw in the F1 generation. These individuals that have only the white flower allele would be white. And so what we would get in this F2 generation is a 1:2:1 both genotype ratio and phenotype ratio of red, pink, and white. So the reason that we know that this isn't blending inheritance is that we get back our original parental traits in the F2 generation. There's nothing that happened to these characteristics, red and white. They weren't irreversibly blended in the offspring. Those gene alleles are still present. We just have to combine them in the right ratios in order to see those phenotypes reappear in subsequent generations. Okay, so that's incomplete dominance. A special case of incomplete dominance is what's referred to as codominance. And in this scenario, the phenotypes associated with both alleles at a given gene are equally expressed and visible in the heterozygote or in the hybrid. And the ABO blood groups are a really nice example of genetic inheritance that illustrate a couple of interesting genetic inheritance patterns. So in some sense they illustrate the dominance and recessive pattern that we talked about previously, as well as the codominance pattern that we are going to describe now. So first of all, let's talk about what the ABO blood groups represent. So this is your blood type, right? You all know if you're type O, type A, type B, type AB blood group, and maybe you know your RH factor, which is represented as a plus or a minus. So the ABO blood group is determined by a specific gene in the genome, that's typically symbolized as the I gene, by the letter I. And what this gene encodes is an enzyme that adds a sugar group to a specific membrane protein on our cells, including our blood cells. So remember, we talked earlier in the semester about the fact that our cells have this extensive carbohydrate matrix that extends up from the surface of the cell. This would be a red blood cell that we're looking at here, and all of this fuzzy stuff that we see projecting up is either sugar groups that have been added onto proteins or sugar groups that have been added onto lipids that are embedded in that phospholipid bilayer. So what the I gene encodes is an enzyme that glycosylates a membrane embedded protein. And this I gene encodes three different alleles. So, well, let me restate that. So there are three different alleles at this I gene that are commonly present in the human population. All right. One of those alleles is symbolized as IA. So the IA allele. The second allele at that gene is symbolized as the IB allele. And the third allele that's commonly found in the population for the I gene is the lowercase I allele. Now, these are slightly different DNA sequences encoding this enzyme that glycosylates a membrane protein, right? And the way that they differ is as follows. The IA allele adds onto what's referred to as a core oligosaccharide or sugar group, which looks like this. So these little spheres are representing different sugars, and your legend for what sugar you're looking at is down here. So the faint pink is N-acetylglucosamine, which we talked about earlier in the semester. The slightly darker pink is galactose. This bright pink is a fucose sugar, and this sort of beigeish brown pink, mauve, maybe we'd call that, is N-acetylgalactosamine. So different sugar groups arranged in different patterns. So this boxed region that I've shown here is a core oligosaccharide. Sorry, it should be an I. Core oligosaccharide. So this is a core sugar structure that's added onto the protein. And then this I gene enzyme is responsible for modifying this core oligosaccharide. So adding something on or not adding something on. All right? So in the case of the IA allele, this version of the enzyme adds on an N-acetylgalactosamine residue onto that core oligosaccharide. And we call that the A-type sugar. All right? So that's the A-type sugar pattern. The B allele adds on a galactose. So we would call this the B-type sugar pattern. The I allele encodes a non-functional enzyme that adds nothing, right? So no additional sugar gets added. Only the core oligosaccharide is present because it was added previously by another enzyme. And that core oligosaccharide is sometimes referred to as the H antigen or H oligosaccharide. But no additional sugar gets added on because this allele encodes a non-functional enzyme that can't do anything with this core oligosaccharide. All right? And so there are three alleles segregating in the population. However, we know that as diploid organisms, we are only going to get two out of the three. In any given individual in the population, you're only going to get two, right? So let's say that you inherit two IA alleles from your parents, right? IA allele from mom, IA allele from dad. What are your blood cells going to look like? Well, you're only going to be producing those membrane proteins with the A-type sugars, right? And so you're going to have type A blood. Let's say that you inherit only the B allele at this gene. Likewise, you are going to be having only the B type sugars present, right? Now, what if you inherit an A allele from one parent and the little I allele from the other parent? Remember that this allele does nothing. It encodes a non-functional enzyme. But you would have functional enzyme producing the A-type sugars. And so if you have this genotype, IA little I, or an A-type allele and non-functional allele, that's also going to give you type A, right? And similarly, if you inherit a B-type allele and a little I allele, that's going to give you type B blood, just as we would have gotten here with the genotype IB. So this looks like simple dominance and recessivity, like we talked about before. The IA allele and the IB allele are both dominant to this little I allele, which is recessive. When you combine these two alleles in the same individual, this phenotype of having no additional sugar added recedes into the background. It might be present, but we see these A-type sugars present on the surface of the cell. So these dominate over this recessive allele. So that's just like the inheritance that we've seen previously. Nothing new here. Simple dominance and recessivity. But what happens if you inherit this from your parents? You inherit an IA allele from one parent and an IB allele from the other parent. Well, in this case, both of these alleles encode a functional enzyme, but each enzyme is doing something slightly different, right? The A allele at this gene, that enzyme is going to be synthesizing those A-type sugar patterns, but the B allele enzyme is going to be synthesizing those B allele type sugar patterns. And so what we're going to see on red blood cells and other cells in the body is both sugars present, and that individual would be referred to as having type AB blood. So this scenario in which the phenotypes and the phenotypic expression of both alleles are fully expressed in the hybrid or heterozygous state, we call codominance. Neither allele predominates over the other. Both are fully expressed. All right? So that's codominance. So that's a new inheritance pattern that we had not talked about before. All right, so what about this business of the rhesus factor or RH factor? So this is a different membrane protein. It's not a sugar group that we're referring to. It's actually a membrane transport protein, and we don't actually know what the function of this transport protein is, but we do know that it looks like a membrane transport protein based on the functional domains that it contains. We just don't quite know what the function is. And this membrane transport protein, I should say, that we refer to as the rhesus factor that's on our blood cells, is encoded by a separate gene in the genome, so not the I gene, but a gene typically referred to as the D gene, right? And at the D gene, there are two possible alleles that we can symbolize as capital D and lowercase d. The capital D allele encodes a functional membrane transporter. Mysterious as it is, we don't know what it's transporting, but it seems to be a functional membrane transport protein. And the lowercase allele encodes actually no protein at all, so not present. So it's a non-functional protein that never gets inserted into the membrane. So we don't get any membrane transport protein inserted. So simple scenario, two possible alleles, and in this case, having even one of this capital D allele, so in other words, having a genotype that looks like this, dominant capital D allele, and anything else, means that your cells do have that membrane transport protein present. And we refer to that as being positive. Yes, you have the membrane transport protein present. So either the genotype homozygous for this functional allele, or heterozygous, will give you that membrane transport protein in your red blood cells. If you have only this version of the gene, this lowercase d allele, then that means that you have no transport protein present in your cells, and we refer to that as being negative, right? So Rh positive and Rh negative is referring to your phenotypic expression at the D gene, where Rh negative means you're not producing that membrane transport protein. It's not present on your red blood cells or other cells in your body. And if you're homozygous or heterozygous for having even one of a functional allele at this gene, then you will express that and you will be Rh positive. So knowing that, we can now actually think about blood types and human blood groups with a little bit more complexity. So thinking about it from the perspective of maybe a dihybrid cross perspective, taking into account possible phenotypic expression resulting from inheritance of both genes. So what I mean by that is let's take an example, right? Let's say that you have an individual who has the following genotypes at the I gene and at the D gene. So this individual is heterozygous at the I gene, so having one IA allele and one little I allele, and also heterozygous at the D gene. And that person goes out, marries, has kids with someone who is heterozygous at the I gene and at the D gene as well. Now, in terms of phenotype, what would be the ABO blood type of this individual? Well, they have a functional A allele. They do have the functional version of the D allele, so they would be type A positive in terms of their blood type. These individuals would be B positive in terms of their blood type. So here's a thought experiment for you to carry out. You have an individual who's A positive here, an individual who's B positive here. Is it possible for them to have an offspring that is O, type O negative? Oh, so actually, I think that I failed to mention that alternate genotype here. Sorry, you probably figured this out on your own, but this was an omission on my part. If you inherit two of these little I alleles, then you have no sugar groups being added to your red blood cells, and that is what gives you type O blood, right? Sorry, nope, not type O, type O blood. So fully recessive, as we talked about before. I'm sure you guys made that leap of logic there, but just to flesh that out more fully. All right, so going back to this example, is it possible for these two people to have an offspring that has type O negative blood? So that is the question. Is it possible? Now, how would we answer that question? Well, as we said before, we could draw out all the possible gamete types that this parent could produce, all the possible gamete types that this parent could produce, combine them in all possible combinations, and figure out if any of those combinations give us this phenotype of O negative blood. That's one way you could do it. That is a time-consuming way, and on an exam, probably not the approach that you would want to use. The far more straightforward way is to think about, if this is the phenotype, what genotype would give us type O negative blood? So what genotype or genotypes, it's possible that there could be more than one, will lead to O negative as a phenotype? So what genotype would we need at the I gene to end up with type O blood? We'd have to be homozygous recessive for that little I allele. What genotype would we have to have with respect to the D gene in order to be Rh negative? We would have to be likewise homozygous recessive at the D gene. So far more easy than figuring out all possible gametes and trying to find a genotype that corresponds to this phenotype in all of the offspring produced is just figure out what genotypes will give you that phenotype and then figure out the probability of ending up with that. So with these two parents, what's the likelihood that they are going to have an offspring that's homozygous recessive at the I gene? So looking just at this I gene genotype first? Well, again, we can draw out our Punnett square. This parent is either going to be contributing the A allele or little I allele. This parent is either going to be contributing the B allele or little I allele. And if we visualize that Punnett square, and I'm drawing it out, but as you get more experience with this, you won't need to draw it out. You'll just have an instant snapshot in your head of what this looks like. 1 out of 4 is going to give us that homozygous recessive genotype of little I, little I. What's the likelihood of ending up with homozygous recessive for the D gene? Well, same thing. We've got 2 heterozygotes here. If we draw out our Punnett square, each parent is either going to be producing the capital D allele gametes or lowercase d allele gametes. Combine those in all possible combinations, and we see once again that 1 out of 4 we would expect to be homozygous recessive. Now we're saying we want this genotype to happen in the same individual as this genotype, so and, right, we want these two occurring together, so we multiply these together, and 1 out of 16 is the fraction or proportion or likelihood of these two individuals having an offspring that's O negative. So just to get some more practice with this, why don't you see if you can figure out what is the probability for these two individuals to have a child that is A positive. What is the probability for them to have a child that is O positive? And what is the probability for them to have a child that is AB negative? And these we will answer in class. So do them on your own, and then we will answer them and review your answers in class and answer any questions that you have about that. Okay, so why is it that this A allele is completely dominant over this recessive allele in terms of phenotypic expression? Why is it that these two alleles, when combined in the same individual, are both fully expressed and neither one dominates over the other? Well, to understand the dominant and recessive phenotype interaction, we really have to be thinking at the level of the molecule, the protein usually, that's encoded by this gene, right? And think about when we have more than one version of that gene product combined in the same cell or in the same organism, how do they interact? Does one override the other in terms of the phenotypic expression or not? Are they both equally likely to have their phenotypes expressed? In this case, both of these alleles, the A allele and the B allele, encoded functional enzymes. There's no interaction between the two beyond that, and each enzyme is doing its own thing, carrying out its own function, which is adding sugar groups onto these membrane proteins that have these core oligosaccharides already added. So having this version of the enzyme present doesn't really influence what this enzyme is doing. They're both doing their own thing. But if we think about having this non-functional enzyme present in the same cell type as one of these functional enzymes, we can see this enzyme is doing nothing, but these are still fully functional. And so their phenotypic expression masks this non-functional expression by virtue of the fact that they're actually doing something in the cell, and this little I alleles enzyme is doing nothing. And so we don't see that nothing phenotype because it's being masked by these other enzymes and their expression. So we really have to be zooming in to the level of the protein and thinking about interactions in that sense. For our rhesus factor, we were actually looking at the presence or absence of this specific membrane transport protein on the surface of our cells. If you have this non-functional allele and you don't produce any protein, then if you do have, as your other allele, a functional version that is producing protein, well, you're still going to have protein present, right? And so this is dominant over the recessive state of having no protein. So having a little bit or half as much is just as good as having the homozygous state, which gives you twice as much. So to flesh this out a little bit more, let's return to an enzyme that we've talked about before, tyrosinase, which is the animal version of our PPO enzyme that we looked at in lab. We know that the tyrosinase enzyme is responsible for initiating the pigment production pathway that occurs in skin, it occurs in hair, and leads to the production of melanin pigments that provide our skin and hair color. So we know that if you have the wild type version of this tyrosinase gene, then you have a functional enzyme. You have a functional tyrosinase enzyme that's capable of kicking off that pigment production pathway. So let's represent that allele, that functional allele of the tyrosinase gene, which is symbolized as TYR, T-Y-R, by this capital T symbol for allele. So this is our wild type normal function allele. But we know that out there in the population, random mutations are occurring all the time, and in some individuals we may have mutations to this gene, right? So let's say, we'll look at two different scenarios. Let's say under scenario 1, a random mutation occurs in that tyrosinase gene that destroys the enzyme's function. So it no longer produces a functional enzyme. Now it produces a version of this protein that has completely lost its function. Maybe it's a mutation that changed an amino acid in the active site. And now that that amino acid is not present, we have no function, no ability to carry out that oxidation reaction. So we will represent that non-functional allele by a lowercase t, right? In some other individual out there in the population, maybe we have, under a second scenario, a mutation occurs that causes the enzyme to be overexpressed or to be overly active. So overexpression or hyperactivation of this enzyme, maybe by causing too much of the enzyme to be present. So disruption in the regulation of transcription so that we get too much expressed. So let's say that mutant allele of this gene we're going to represent by a capital T with an asterisk. So this is the hyperactivated allele of the tyrosinase gene. Now, to think about the dominance and recessive relationships of these two mutant alleles in the context of an individual that also has a wild type allele. So in other words, let's say in individual number 1, they are heterozygous and they have this genotype. They have one wild type allele, normal allele, and then they've inherited one allele that does nothing. What would we expect their phenotypic expression to be, given that genotype? And on the other hand, what would we expect the phenotypic expression to be in an individual that has one wild type allele and one hyperactivated allele expressing too much or overactive tyrosinase enzyme? So again, take a moment, pause the video, make your predictions, think about what you think would happen under either of these two scenarios. Okay, so what predictions can we make? Well, let's think about this. This allele is the normal allele, right? It has normal function. It's carrying out its job. It's initiating the pigment production pathway as usual. It's converting tyrosine to one of the quinones, right? Kicking off this pigment synthesis pathway. Well, what is this allele encoding? Nothing. No function, right? It's doing nothing. In the context of a cell that contains both of these versions of the tyrosinase enzyme, this one's going to be doing nothing, but this version of the enzyme is going to be carrying out its normal activities. And so we might expect that the pigment production in this cell is going to be normal, because we do still have functioning enzyme in the cell initiating that pigment production pathway, right? And so if that's the case, then the only way that we would see the expression of this lowercase t allele in any individual is if they inherited two bad copies, right? This would be recessive in its phenotypic expression, and this would be dominant in its phenotypic expression. When we combine the two in the same individual, we see the phenotype associated with this dominant normal allele. We still get pigment production taking place. Okay, what about the other scenario? What about the other scenario where we have both the normal enzyme present in the cell, but we also have this hyperactivated or overexpressed version of the enzyme? Now, if we think about this, this allele and its gene product is going to be carrying out normal pigment synthesis in the cell, but what is this allele going to be doing? It's going to be producing too much pigment, right? Overproduction, either because we have more of this enzyme than we're supposed to have, or it's hyperactive and it's catalyzing too much pigment production. And so in that scenario, does it matter that we have this normal function tyrosinase doing its thing as it should? No. The phenotype that we're actually going to see is this hyperactivation or overexpression phenotype. And so in this scenario, that mutant allele would actually, be dominant over our, oh, it doesn't give me that, that's right. That mutant allele would actually be dominant over our wild type allele. So this one would be recessive. Okay, right? So in general, when we have a gene mutation that changes the function of the gene, right? So we get a new allele produced by random mutation, and that allele results in a loss of function. When that's combined with a normally functioning allele, that's going to be recessive to that normally functioning allele. On the other hand, when we have a mutation that leads to a new function or overexpression of the normal function, those mutations are typically dominant in the genotype context of being combined with the normal allele. So gain of function or loss of function mutations are associated typically with recessive inheritance patterns and expression patterns and dominant inheritance patterns or expression patterns respectively. Okay? And that's in general. Not 100% of the time. You actually can have loss of function mutations in some scenarios that will be dominant over the wild type or normal allele, and typically that's when there's a complex that's formed between the two, and so one essentially can poison the other. But that's not typical. That's not the prevalent or most common scenario that we would see. All right, so let's think back to our cell signaling unit and try and apply this idea of recessive mutations versus dominant acting mutations. So let's say that we're looking at a receptor tyrosine kinase signaling pathway, which we talked about before. We know that normally, under normal scenarios, we have these individual receptor tyrosine kinase receptors, and when each receptor gets a ligand bound to it, we see this dimerization taking place, this cross-phosphorylation that happens where we now have phosphorylated sites on the intracellular surface of this enzyme receptor, and those are now binding sites for cellular proteins. These are cellular signaling proteins that bind. They're activated by the kinase activity of the receptor, and they go off and diffuse into the cell and trigger specific cellular responses, typically via these kinase and phosphatase signal transduction cascades that happen within the cell. So thinking about recessive acting mutations and dominant acting mutations in this sort of a signaling pathway, could we postulate different types of mutations and disruptions to function that might act in a recessive fashion or that might act in a dominant fashion? So keeping in mind our loss of function, gain of function. Think about that, and this is another thing that we'll talk about more in lecture. So when we have our genetics problem solving activities, we'll return to this example and think about that. But between now and then, go ahead and spend some time thinking about this and just hypothesize some mutations in different components of this signal pathway that you might expect to act in a dominant fashion or in a recessive fashion. What would be the mutation in what component in the receptor, in the cellular response, in the ligand or upstream, and why do you expect it to act in a dominant or recessive fashion?